In this video, I will teach you how to calculate the size of DNA fragments you will get after performing a restriction digest. Restriction enzymes cut the DNA at specific locations. Some do it at one location, some do it at two. Because this is just an example, we're going to say each restriction enzyme cuts it at one location. So enzyme 1 here cuts the plasmid at 85 base pairs after the origin. Enzyme 2 cuts it at 760 base pairs. The whole plasmid is 1,644 base pairs. By the way, all these numbers in plasmids and enzymes, they're just made up. There are obviously real plasmids and enzymes, but I made my own up to avoid copyright. So now to calculate it, what you have to do is you have to first take away 85, which is the smaller number where the first one cuts, and subtract that from 760, which is where the larger enzyme cuts. That will get you this fragment length in base pairs. And that's an easy 675 base pairs there. Now for the other one, you simply subtract 675 from the total length, which is 1,644. And we're only dealing with two enzymes here. And that equals about 879 base pairs. Now, suppose you have multiple plasmids, and this is for an application such as site-directed mutagenesis. So one of these has a mutation, let's say, at the restriction site. Restriction enzymes only cut at specific locations, so if there's a mutation there, it might not cut depending on the enzyme and depending on the mutation. So let's say enzyme 2 doesn't cut. What fragment length will you get here? Well, simple. Enzyme 1 will still cut, but enzyme 2 won't. So you'll just get one long straight strand of 1,644 base pair DNA. And that is actually a way that you can detect whether site-directed mutagenesis has worked or not, because the wild type and mutant give different DNA fragment lengths in the restriction enzyme digest. And this difference can be detected through gel electrophoresis, which separates DNA fragments based on size, length, and base pairs.